So today in this demo, I will discuss and show you how to use the Microscopy Image Analyzer Tool that we've been building. Right. So basically, it is designed to simplify and speed up the analysis of microscopy images and providing you the accurate and clear results for your experiment. So it goes beyond just measuring the cell size or shape. You can also classify the cell types. You can track population trends, and then you can also use it for advanced techniques like the principal component analysis to interpret your data. All right. So before we delve into it, let me just let you know. All right, the this tool is literally designed for both phase contrast and fluorescence images. So, and it also supports other data sets like the TIFF. So you can literally upload a folder and then it's going to work the same way. But that part is not really completed yet. All right. So so I'll walk you through each tab: the import tab, the export, the population. We have the morphology tab, and inside the morphology tab we have the scatter plot, the PCA plot, which is the principal component analysis plot. Then we also have the metrics table as well, showing the morphology metrics analysis, right, that helped us to generate the plots. And then we also have the morphology time tab as well. So basically, I'll walk you through each tab in this application and we can explore its features step by step. Let's delve into it right now. So uh, the first thing we'll do is we'll begin with the import tab, right, which is where we load our image data into this tool. Right, when you load it, it appears here. And right, so you can also load directly from the folder, which is the TIFF images that you have in the folder. When you click on this, which means you can select each folder, and since I've checked this now, so if you want to load anything, it doesn't load the file. You have to select the folder before you can do this. But right now, this process is not working yet. I mean, I would have tried this and then open, but it's not gonna work because the part is not completed yet. But definitely, it's a work in progress. So right now, we are going to be uploading the data sets that we have, the ND2 file. Right, so you just click the import tab, right, and click the button to select file or folder, right, and then once the data is loaded, you are going to see the information about the data sets right on this tab, on this image label, and this is what we can see here. Right, so then after this, I believe you can be able to see the position, the what's it called, the toggles here. You can toggle between the time frames and the position as well. You can also see the channels if your data set has fluorescence. If it doesn't have fluorescence, obviously you're not going to see any channel here. So because this is just phase contrast data set, we are not going to be able to see all of this information about the fluorescence, which is the channels. All right, so let us go into the next tab. Basically, all of these things we have here is just these are just further information, right, more like analyzed information of time. So we have 49, we have like about 0 to 49 time frames, and then the position is just from 0 to 9 from 0 to 8. Basically they are just 9. And then these are the coordinates, basically the x coordinate and the y coordinate. Then also this is just if you want to basically only test within a certain time frame, right, like a certain range of time frame. So after setting this to 7 right now, you can see what happened to this toggle. Now we have from 0 to 7. Same thing applies to position and the channel, but right now we don't have any channel, so that is why we don't have any drop down here. Also the x coordinate is fixed and also the y coordinate is fixed. All right, so now let us move on to, after we've processed the data, now we can move on to explaining the next tab, which is the export feature. Currently, what we have for the export feature, right, it's for us to okay, there's a particular button here which is called slice and export. So basically what this is doing, right, is it slices these images, this image, based on the time frame that you set for it, and it slices it. We can text that and experiment it. So we'll slice it and export it as TIFF files. Okay, so let's see, let's put it here. Okay, then save it. Alright, so the slice data has been stored into the folder. Let's go ahead and check, and then we can confirm. Alright, so basically this is what we have. We have the slice data converted into TIFF file, right, from 0 to T2, from T0 to T2, and from P0 to P2. So basically this is just an experiment we're trying to work on. We're testing something, but I mean, we'll come back to that. This is not really an important feature on the tool right now, but it is a work in progress. Basically everything that requires the T folder being uploaded, and the images are aligned and all of that, is currently a work in progress, and it will be completed as soon as possible. All right, so this export feature basically is just to save the process data, but right now it's not saving the process data entirely. Right now it's just basically saving the images, is processing the images and converting it to TIFF images, right? So instead of saving it, basically we are trying to save it as ND2 file. It processes it this way. It's not really doing much, but it's just exporting it as a TIFF file. But the improvements that is currently going on this particular feature is for us to be able to process the images. Let's say segmenting the images, and then when it's segmented, you can come here and export the data, or basically the what's it called, let's say your scattered plot or whatnot. You can be able to like export at any point in time the results that you are getting from every analysis that you are doing. For instance, you are doing your population trend analysis, you can be able to export it from this particular trend as well. From this morphology, you should be able to export it directly. So this export feature is currently being updated, right, as well. So but let's just get into the more interesting part of the tool, and then all of these little fixes that are currently work in progress at the moment. So as at the time of doing this demo, which is going to be a version 1 of this video, right, these are the things that we have working, right? So all right, so we have this view area, right? So this view area where you view the images, you can be able to classify the cells and segment it. Then these are just raw images. This is just the raw data, where you can be able to see the all the cells, and but you can literally not see the cells very well, because we still have the background noises around, and then they have to be cleared. So to do that, first we segment the cells, right? When you click on this segmented button, I mean the radio button, when you click on it, the process begins. So the segmentation process has started, and you can see that this what's it called, it keeps loading here. There's a loader here, so you just have to be patient for a little, and it's segmenting at time frame 4 and position 1 actually. So we just wait on for a while. So like I said, you can use these sliders to move through the time points, position, and channels, and that is what I did to set it to time frame 4, position 1, and then I click the segmented radio button in order to view the segmented cells basically, so that I can see the cells clearly. Let's just wait for a little while. All right, so now we can see all the cells that we have here, right? So this after processing the images, right, then we click to segment it. Now we can see that it has cleaned the entire images and then cleared out the noises and every other thing that is preventing us from seeing the cells clearly. And this is what we have here after it has been segmented. This is the segmented image. So basically what we want to just do, we have to probably classify the images to see which images to see the what's it called, each class of images, whether they are elongated, whether they are deformed, or whether they are normal or right. So if you click classify cells, right, so it does this. So currently what we have here might not be very clear, but if you right click on this view area and you click to save the annotated images, basically it's just annotating it and setting the algorithm is just to set it has been configured in such a way that each cell has been accurately classified. So we have the elongated images, we have the deformed images, and they are set in different colors. But right now it's just bounding boxes around it. But it is currently a work in progress in such a way that instead of having bounding boxes around it, we just need to like color each segmented cell, right, based on their classification. So let us just export these images now, and then you can see it clearly after it has been exported. Okay, I think I'm saving inside demo folder. Let me just do it inside dataset. Alright, so that has been saved, and then we can check it later. But let's just check another interesting feature. So now after this cell is annotated, as long as you've segmented the cell, then you can be able to run at this specific time frame, right? Then you can be able to run the classification, you can be able to run the morphology time. Basically what we see here now, the morphology has been set, right? Since you click to classify the cells, right, so the classification it sets the morphology and it also plots the table. Right now we have the morphology table, and you can see that at every, so it's currently another work in progress, right, where in such a way that when you click on each row, so basically these IDs are each ID of the cell, right? So ID1 is currently somewhere in the cell, ID2 is in the cell, ID3 is in the cell. So the table is plotted with the accurate area, perimeter, equivalent, and every other metrics that we use to calculate this, to analyze these cells, I mean to classify the cells. And at the end of the day, based on the PCA analysis, right, we were able to calculate the morphology, right, and we're able to discover the elongated cells and the deformed cells and the normal cells as well. All right, so this is what we have here now. So basically the idea we're currently working on is we are supposed to be able to click each point and then it highlights on this image, right? Like you click this point, right, and then you see the particular image it is. So you click any point here, you see the image. So you can see here that we have clusters of elongated images around here, and then we have some little clusters of deformed image around here, and we have some little clusters of normal images around here. So basically this is what the PCA does. It helps us to figure out the clustered images, right, of different classes, and it merges them together, it pulls them together, and then we can be able to do further analysis of some of these things, right? So also the metrics table gives us a clear idea, and we can export this data here for further analysis, into other tools, the biofilm queue and the other tools like that, and then you can use it for further experimentation. All right, so the feature to export it right now is not completed, but as at the time of making this video, is not completed, but it will be as soon as possible. All right, so that's for this. And then the morphology time tab as well. This is just to process the metrics, morphology metrics over the time, right? That's just the feature for this, right? But we might have to wait a little while because it has to compute for all the time frames from 0 to, but right now we have to set it from 0 to 6 because of the consumption of time, right? And for the delay, to prevent the delay. So but the idea right now is it computes from 0 to it computes from 0 to the to t minus 1, which is the time frames. It computes for all time frames, right? So basically we have how many time frames here? We have about 7 time frames, I guess. So because from the imports tab, we were able to set the time frame limit to 7, so it's from 0 to 7. So the idea is supposed to compute from 0 to 7, right? But if we didn't set this, of course it's going to be from 0 to 49, which is the time frames computed in the dataset. So this is what we have here. Now it's keep loading, and then we can see a progress bar down below, and this is what we are going to use to track the progress. Here we can see it's tracking the progress. Now it has computed for T1, it's going to compute for T2, T3 till T6. We have to set it to T6 so we can be able to experiment it further. When we push this to production, and then you want to access it, it's going to take the original time frames. All right, so while this is going on, let's just test the population as well, right? So for the population tab, you can analyze the intensity threads across your data set, basically. So this is just like analyzing the intensity threads, actually. So it doesn't really require time frame because it is done at a specific time frame. So this is at T4. So this is, you can adjust the position here, right? So yeah, there are some questions that were asked during the first meeting that we had where we demoed this, and that was where Ahmed asked a couple of questions, and the truth is they are currently work in progress right now, and it will be deployed, or will be deployed and be completed, and probably a version 2 of this demo will be done right next semester, right? But this is going to just give you a good idea of what the tool does, right? And when the next version comes, it gives you so it makes it easy for you to work your way around it. So to test this right now, then you just need to like move this slider, right? So you just use this position slider to select a specific sample region. So the position that we set to position 1, can set it to position 2, position 3, position 4. I believe you just have 4 positions here. Let me see. Yeah, so the position was set to 4 here. So that is why it's working based on the position that was set at the import tab. So originally, if it wasn't set, it's going to take the entire positions. All right. So now the tool has calculated the average pixel intensity across all time plots. So it has calculated for all time plots from 0 to basically to set the current time plot is 0 to 7, right? So that is what is happening right here as well. So it's calculated from 0 to 7. So initially, if it wasn't from 0 to 7, it's going to be taken, if it wasn't set to 7 here, it's going to be taken from 0 to 49, right? So that is what we have here. And that is exactly what this tab is working on, is doing. So this feature is great for can help you to track population wide changes in your intensity over time, right? So, and whether you are working with fluorescence or phase contrast datasets, it works the same way. So I believe you're good with this pixel intensity tab. All right, so this is almost done. Now we are trying to calculate the area metrics over time, and we are almost done with this. Once this is done, then we can proceed to see the plots. All right, so there's one last thing that I want to show you. So we have this segmentation models, right? So this, as you can see, is still a work in progress basically. So we have model A, model B, model C. The idea is for us to be able to switch between different models and compare their results. So basically, we're not supposed to see error difference that is so large, right? Just so we can be able to the uh, um, confirm the results that we are getting from each model, right? So that's why we have this. And currently, the model we are using is the cell post, and which uses the UNet model, right? So that is currently what we have to generate this, beautiful results that we are getting here. But quite a lot of work is still going on, and to give us more accurate results. All right, so this has completely computed. So we have from 0 to 6. Actually, this is just hard coded, right? The 0 to 6 is hard coded. So we just did that just so that we can be able to test accurately, and because of the time it takes to compute. All right, so that is what we have here. And now we have the morphology time graph, and we can see it here. All right, so this is basically everything that we have for this tool, right? So, and in the SOP provided, we stated there that you should include some details, right? Some information about your data set, which is quite important for us in the experiment, in improving the tool. That's one. Also, some requirements from the biologist to be provided alongside with your experimental data, so we can these are things that can help us to improve the system in such a way that it can give you as many results as you want, right? All right, so this is everything we have here, and thank you for if you have any questions or suggestions or whatnot, you can always reach out on Slack, and then we discuss further there. All right, so I just hope this tool works fine for you, and I hope you understood every thing that has been said in this demo. All right, so basically, when you're analyzing, whether you're analyzing single cells population or the entire data set, this tool can actually help you extract meaningful results from your images, meaningful processed results from your images. And then it is always a work in progress. So basically, we are always working on it. Like, we are always adjusting it to suit your to suit whatever you want and the kind of results that you're expecting. All right, thanks for watching, and yeah, happy experimentation. We will always be available if you need any question, if you need any clarity on everything that's been discussed here. Thank you.